The man. The man is mentioned in Quran chapter 76. In fact, chapter 76 of the Quran is named Al-Insan, the man. Human begins from a drop of mixed fluid, nothing yet worth mentioning, where a single sperm carries 23 chromosomes, not pairs. Within that sperm, genes like p53 silently prepare to guard the future embryo's genetic integrity. Is there not a period of time when each human is nothing yet worth mentioning? For indeed, we alone created humans from a drop of mixed fluids in order to test them, so we made them hear and see. Al-Insan 76 verse 1-2. Human reproduction, from the formation of gametes to the earliest stages of embryonic development, is profoundly influenced by the TP53 gene and the critical p53 protein it encodes. Often hailed as the guardian of the genome, p53's role extends beyond its well-known function in cancer suppression to actively police the integrity of genetic material, including the unique chromosome 23, the Y chromosome, of sperm, at every step towards successful fertilization and beyond. During the demanding process of sperm production, known as spermatogenesis, the TP53 gene, located on chromosome 17, is indispensable. It acts as a vigilant quality control checkpoint, scrutinizing the vast amount of DNA replication and meiotic division occurring in developing sperm. If significant DNA damage is detected, whether on autosomes or the sex chromosomes including the Y chromosome, chromosome 23, p53 can trigger cell cycle arrest to allow for repair or, if the damage is too severe, induce apoptosis, programmed cell death. This crucial mechanism ensures that only sperm carrying intact and healthy genetic material, including a fully functional Y chromosome essential for male offspring, are allowed to mature. Without this vital surveillance, compromised sperm with fragmented DNA or chromosomal aberrations, potentially including Y chromosome deletions common in male infertility, could proceed to fertilization, leading to failed pregnancies or developmental issues. The influence of TP53 extends directly into the momentous event of fertilization and the subsequent delicate stages of early embryonic development. Once fertilization occurs, the newly formed embryo, a fusion of genetic material from both parents, continues to be under p53's watchful eye. If the combined DNA from the sperm, including its X or Y chromosome, and egg contains errors or aneuploidies, p53 can orchestrate the elimination of compromised embryonic cells, acting as a safeguard against developmental abnormalities. Beyond this intrinsic embryonic quality control, p53 in the maternal uterus plays a pivotal role in implantation. It regulates the expression of key factors, such as leukemia inhibitory factor, LIF, which are essential for the uterine lining to become receptive to the implanting blastocyst. Genetic variations in TP53 have been associated with implantation failure, highlighting that the success of fertilization is not solely dependent on gamete quality, but also on the maternal environment influenced by this guardian protein. Thus, from the initial production of viable sperm, with their intact chromosome 23, through the meticulous quality checks of the early embryo, to the critical stage of uterine receptivity, TP53 stands as a fundamental player in the intricate biological symphony of human reproduction. The name p53 comes from the combination of the letter P, which stands for protein, and the number 53, which refers to its apparent molecular weight of approximately 53 kilodaltons as measured during early laboratory experiments. Interestingly, the actual molecular weight of the p53 protein based on its amino acid sequence is closer to 43.7 kilodaltons, but it migrates more slowly in gel electrophorus due to post-translational modifications, making it appear heavier. This discrepancy is common in protein analysis, and led to the protein being universally recognized and named p53 in scientific literature. Fun math. The chapter of Al-Insan, with its 1078 letters, holds a deeper secret. Human begins from a drop of mixed fluid, where a single sperm carries 23 chromosomes. Genes like p53, 43.7 kilodaltons, silently prepare to guard the future embryo's genetic integrity. Within its text, the numerical sequence 23,437 acts as a hidden blueprint. When its values are brought together in a specific order, their sum resolves to a total that is a perfect mirror of the chapter's letter count, 1078, standing as a silent testament to the beauty of creation. 234 plus 343 plus 437 plus 23 plus 37 plus 4.